Good morning. I'm Tyler Martin, and here's the latest from Spectrum Autism Research. Spectrum of variants contribute differently to autism traits. By Laura Dotaro. Two new studies untangle how various classes of genetic variants underpin the vast differences in traits seen among people diagnosed with autism. The studies were published yesterday in Nature Genetics. The fundamental question behind this is heterogeneity in autism, says Varun Warrier, a postdoctoral researcher in Simon Baron-Cohen's lab at the University of Cambridge in the United Kingdom and an investigator on one of the studies. The presence and intensity of core autism traits and co-occurring conditions vary widely among autistic people. The new studies from largely independent teams sought to unravel how different categories of genetic variants, rare, common, inherited, and spontaneous, contribute to this heterogeneity. Though the two sets of findings conflict in some ways, potentially because of methodological differences, the papers add to the evidence that common and rare variants contribute to autism's genetic architecture differently, says Yufeng Xin, associate professor of systems biology at Columbia University, who was not involved in either study. When we say different, it's not black and white, Xin says. They overlap, but it seems like qualitatively they have different contributions. Warrier and his colleagues analyzed genetic and behavioral data from 12,893 autistic people. The data came from the Autism Genetic Resource Exchange, the Longitudinal European Autism Project, the Simons Simplex Collection, and SPARC. The Simons Simplex Collection and SPARC are funded by the Simons Foundation, Spectrum's parent organization. The researchers combed the genetic data for disruptive de novo variants and used previously calculated polygenic scores, tallies of common variants linked to a particular trait, for autism, attention deficit hyperactivity disorder, ADHD, schizophrenia, IQ, and educational attainment. Then they looked for associations among the variants or polygenic scores and 19 behaviors central to or often associated with autism, including social difficulties, insistence on sameness, and problems with motor skills. Among the findings, high ADHD polygenic scores are associated with increased non-social autism traits such as repetitive behaviors. High educational attainment scores are associated with high IQ and few core autism traits. And high schizophrenia scores are associated with adaptive behavior difficulties. Disruptive de novo variants, however, were not associated with core autism traits. And within a subgroup of 3,052 autistic people, the 325 carrying such mutations have a lower autism polygenic score than those without them, a trend not seen for polygenic scores for schizophrenia, educational attainment, or IQ, and also tended to have co-occurring developmental disabilities. Those with high autism polygenic scores, on the other hand, have few such conditions, a surprising finding, Warrior says. Autistic girls are more likely to harbor a disruptive de novo variant than autistic boys are, the team found, but the mutations occurred entirely in genes linked to developmental delay rather than those linked to autism. It's possible that rare de novo variants contribute to higher odds of any neurodevelopmental condition, Warrior says. Inheriting additional common variants could then lead to autism. All this paper is saying is, heterogeneity exists and heterogeneity influences your genetics, Warrior says. We need to take that into account when studying genetics of autism or indeed any sort of developmental condition. In the other study, a separate team led by Jonathan Sabat, professor of psychiatry and cellular and molecular medicine at the University of California, San Diego, analyzed a combination of whole genome sequences, exome sequences, and genotype data from 12,270 autistic people, 5,190 non-autistic siblings, and 19,917 parents. The data came from a University of California, San Diego cohort, the Simons Simplex Collection, and SPARC. For each participant, the team calculated 3 scores: a common variant score that reflects variants linked to autism, schizophrenia, and education, a rare variant score, which includes de novo missense and loss of function mutations and inherited loss of function mutations, and a combined score that encompasses all 6 factors. People with the highest combined scores had 2.2 greater odds of having autism than people with the lowest scores. For the first time, they actually calibrate the risk from rare variants and put them together with the common variants and show it's largely additive, Shin says. Autistic girls and women had significantly higher rare and common variant scores than boys and men with autism, the researchers found. In another analysis of gene trait associations, people with a high autism polygenic score or a de novo loss of function mutation are more likely to have social difficulties, Sabat's team found. The finding conflicts with Warrior's results showing no link between de novo mutations and core autism traits, Shin says. Warrior's group analyzed de novo loss of function and missense variants together, whereas Sabat's team analyzed them separately, which could explain the difference. This is an extremely important question, Shin says, and future studies should look at individual genes or gene groups. None of the associations between genes and traits varied strongly by sex, suggesting genetic factors act similarly on autism traits in boys and girls. Sabat did not respond to requests for comment. Autistic people with disruptive de novo mutations have fewer common variants than those without such mutations, according to the analysis, though the result was not statistically significant in girls. The researchers also defined a set of 125 genes linked to autism via rare variants and identified 114 genes strongly linked to autism via common variants in a previous large genetic study. Genes from both sets are more highly expressed across the cortex and during fetal development than a random control set of genes, according to comparisons with 2 brain databases. Only the rare variant genes, however, showed higher expression, double that of controls, in excitatory and inhibitory neurons. Spark is expected to release a much larger dataset in the next year or 2, which will dramatically improve the accuracy of estimations of genes' contributions to autism, Shen says. These are important questions, but it's not the final answer yet. That's all for today. Check back on Wednesday for more content from Spectrum Autism Research, or go to spectrumnews.org.